everyone, and welcome to our interview series marking the bicentenary of Gregor Mendel. I'm Asta Vatsyan, a PhD student in Dr. Vinod Skaria's lab. Today we have with us Dr. Vani Brahmachari, who, after a long tenure, has recently retired from B.R. Ambedkar Center for Biomedical Research, Delhi University. In her long and illustrious career, Dr. Brahmachari has worked extensively in the field of epigenetic regulation in disease and development. She has a significant body of translational work, creating huge impact in diseases such as Fragile X syndrome, coronary artery disease, and childhood leukemia. Welcome, ma'am. It's an honor to have you here with us today.
My pleasure.
Ma'am, you've made significant contributions to science and medicine through your work. Ma'am, how do you think Mendel's discoveries have been a part of your work, or how have they impacted your work through the years?
My work didn't have a direct sort of, I worked in the area of molecular genetics more than, pure Mendelian genetics. But if you look at the scenario, ours was majorly a hypothesis-driven approach for research, not in the terms of the big data. More recently, over the last 5 years before I retired, we started looking at large data. The interest came in genetics mainly during my master's course. I should acknowledge that the teachers, especially one professor, R. Jayaraman, he used to teach us molecular genetics, and especially the bacterial genetics. And the whole thing was looking so, logical. And you would go to every experiment that led to various discoveries in terms of the, simple things like lack of ground. So these experiments are so ingenious and then designed to give you a unique outcome. So it was very impressive in that sense. And after that, when I joined for PhD at IISc, I worked in the area of mycobacterium, basically to look at the modified bases in them. And the idea was that whether it would be targeted as one of the targets, a far-fetched thing, which did not sort of, my work did not consist of that. But then we did the sequencing of the transfer RNA. That was early sequencing that was done. And it was an RNA sequencing exercise, which I did in collaboration with National Institute of Cancer at Tokyo. And after that, my interest in modified bases extended to modifications in DNA. And therefore, I got introduced to genomic imprinting and epigenetics. Basically, with Professor T. Ramakrishnan, with whom I did my PhD, and then subsequently, epigenetics came into my interest by my association with Professor Sharat Chandra at IISc. And thereafter, we have been looking at various kind of modifiers. And more recently, of course, the Fragile X work was one of the things, when Fragile X inheritance was looked at, it was peculiar in the sense it would skip generation, and expansion was happening when it is passed through the female rather than male. So then it looked as though that there is some kind of an epigenetic regulation at that time. And while we were at it, it came out that it's a triplet repeat instability that happens, and therefore there is expansion and you see the disease. Of course, it behaves like a typical X-linked disease. There is no problem about it. So then we were looking at the mechanistic aspect of it. In the case of mice, the same gene is there with a very high similarity to the gene, and but then it doesn't do that expand through such expansions. And therefore, we created a transgenic model to bring in certain elements which modify the chromatin, basically. And that yielded a result that we had a cassette like situation wherein we had sequences which can eliminate nucleosomes and therefore promote, looping out and things like that. And then that promoted the expansion of triplet repeats. And by then the genome had come, human genome had come, and the number of epigenetic modifiers, part of the machinery, were very few at that time. There was lots of them known in Drosophila, quite a few of them, but then not every homolog or in the human head was known. And therefore, our search became that we are to look at the genome and then use certain features of the various enzymes, as well as use a novel approach through the PL host. I don't know whether you've heard of this. This was Dr. Devashish Das and Dr. Brahmachari's, they made a software, and I won't get into the details, wherein you sort of create a peptide library and look for identity between peptide libraries of two organisms and pick up those which have identity in reasonably long, 7 and above amino acids. So with that approach, we picked up a couple of candidate sequences, which could be coding for epigenetic modifiers. So when we identified this, that's where we moved to Drosophila, where we did some amount of genetics, because we could see the genetic interactions of the gene and so on. So after that, our approach basically has been to use model system to do the functional analysis of genes, which are the homologs, which we discovered in human by the genome sequence analysis. So that has been roughly the direction of the research. I don't get into every detail, but then it was interesting that this observation from our lab is part of the NCBI annotation for the gene. So that was gratifying. And thereafter, I have been involved in teaching genetics also for post BSc students. That is when they come for MSc. That's one of the things. Yeah. What else do you know?
Ma'am, you have taught genetics, and definitely for researchers, it is extremely essential to know about genetics. Do you also think it's important for the common man to know genetics? How the knowledge would actually help the common man?
See, the thing is, genetics affects everybody, family and inheritance and all that. So this, by just a general knowledge, people have an interest in this area, right? And especially if there are cases of friends or relatives or one's own family, which has some genetic disease running through, then it becomes very important that they know what are the risks of having kids and what kind of genetic background would be preferred in order to avoid getting the, that passed on from generation to generation. For example, thalassemia, which is very common. So these are the kind of things which you want to educate the society so that that is avoided. The birth of such, children is avoided consciously, not by, by conscious matching of individuals. So if you avoid the marriage between two carriers, right, your chance of transmitting through generations is reduced. So in that respect, genetics. Of course, in the COVID times, you can see that everybody is talking about RT-PCR, variant, and so on, right? So it has elicited a natural curiosity because of the nature of the subject and how it affects every organism on the surface of the earth. So that's always there. In the one of the things with genetics, my experience of teaching students used to be that there was a fear of genetics, at least in some of the students. Um, and that comes from the fact that genetics was loaded with jargons, terminologies,
Right.
penetrant, called dominant, recessive, whatever. Dominance, recessive is understood in Mendelian terms. He simplified it and explained, but this is not so simple as, pure dominance, pure recessive. You may not find many cases, especially in the human instances itself, with the whole genome and copies of the genes and things like that. So the terminology is a major sort of uh, barricade for students to sort of take to genetics, at least for some of them, not all. But once the molecular basis of some of these could be explained by regulation and so on and so forth, then it became much more easier, right? The terminologies could be replaced with more precise terms which indicate a biochemical function rather than, saying, for instance, terms like penetrance and so on. If you look at one of the, very popular books on human genetics, Vogel and Budulski, some of these phenomena like penetrance, incomplete uh, inheritance or penetrance and things like that. So they say in the earlier editions that these are terminologies of our ignorance, because we don't know what is a molecular basis. So you describe it, just to have a common terminology to describe the phenomenon you choose, but it does not have any clue for the mechanism involved. But then today we know there can be multiple mechanisms which may be the reason for lack of correlation between the genotype and the phenotype, right? Which is what penetrance is also. In the population, you may not find the same phenotype in individuals who have the similar gene mutated and things like that. So that is one thing that in the teaching, if you bring in the molecular basis, then it becomes much more interesting for the students, and the enigma is sort of made a little more transparent.
Right. Right. Ma'am, you mentioned, for instance, what happened during the pandemic, how people became more aware of the scientific terminology and just basically more aware of the science behind what was going on. Do you think, ma'am, that with increased knowledge, let's say, of genetics and the principles of genetics, would the healthcare policies also end up being shaped differently, maybe by the knowledge of genetics, if more and more people become aware of genetics and the principles involved?
Yes, it should make a difference because it will be more scientifically dealt with. And I think recently, a couple of days back, Soumya Swaminathan's own words were there that policies and public awareness of genetics should be consciously brought in. I know WHO was taking some decision of that kind. So it is, I think, useful in terms of understanding diseases and also getting rid of some of the attaching stigma to people who have any, any problems with their biochemistry or basis of genetics or whatever it is. But then that kind of stigmatization will certainly decrease. And also when you tell them that, one has to go through a sort of a genetic analysis if possible before you decide to have kids or things like that, then the total burden on the society, disease burden on the society can come down, and the suffering also can come down. So I think overall, any knowledge would make a society much more rational and, much more logical. And so knowledge of genetics, especially that it touches so many disease scenarios, I think it's very important that public awareness should be brought in, and then it will have a positive effect. Not to complicate the thought process, but making it palatable to common people, and you will see a major impact if that happens.
Right. Ma'am, what are some of the future steps or advancements in the field of genetics that you think are coming in coming era of science?
See, there is a deluge of data right now. So much of data coming in and so on. So, but then all of it relating to the phenotypes, what it does is not clear to us. We are sort of lost in the data. Now, as you see, organizing the data with your own experience, organizing the data itself is a major, major job, right? Yeah. Depending on the questions that you are addressing. That is important. And then finding the functional elements of these various genes or sequences and so on. Now, this is a very daunting task, though, over the years, everybody has taken a reductionist approach, most of us, and tried to decipher the function of the genes. But we increasingly realize that there are complex networks that work, that interaction networks between the gene products and genes and non-coding RNA and so many other factors. So collating and putting them together is a major task that is being approached right now. And also functional analysis with reference to using other systems also has to be looked at and will be looked at. And there is so much of effort humanizing, organ growth on animals and things like that. But right now it looks like an impossible task to people like us who have, dealt with only one part of the big jigsaw puzzle, right? But putting together this jigsaw puzzle and finding function for understanding the function of these genes and sequences in any genome, I think that is a focus which is already in progress, and we need new approaches to address it in a more collective sense. So we are collecting data and data and data, but then there is a time in which that has to, it needs some kind of an extreme situation or a cooperation in order to put this picture together. But it may be that I am saying something which looks impossible today, but will be made possible several years later. That's true. New things will come, new methods of looking at it will come. And I'm sure people will be able to, will understand these things. So basically that is the direction, but I think still these, what we call as model organisms, right, are relevant for genetic research, even though we come to know so much about the human genome or in the higher organism genomes and so on. So it's a combination of certain amount of reductionist approach and in the context of the large data that we'll have to understand what is being deciphered in terms of the genome content and all the functions of the genome, the interactions. That's a massive effort.
Yes, ma'am. Ma'am, you said new things are coming up, new people are coming up. On that note, I would like to ask you my last and final question for today. With the incredible experience that you have, and especially with the teaching experience that you have, is there a message you would like to give to new and upcoming researchers as to how they should proceed in life, how they should proceed with research? Anything that you would like to share with all of us?
See, one of the things is just to develop an analytical ability, right? Our education system may not provide you that all the time, but then there, one has to develop that, and that would be a major, that will contribute majorly to one's own growth. And also asking questions. This is one thing that, uh, our students are very hesitant about in terms of, and also I think as teachers and mentors, there has to be much open-mindedness and self-motivation of the students, and they should be able to ask whatever it is. It can be contradictory to what we think or what is believed today, but then take up a bigger problem and address those and do an honest research. And that is the most important, I think, that you have to keep it sustained. I know there are a lot of disappointments, but it has to be sustained. And exposure. Exposure is a very important part of science. Listening to lectures, reading extensively, which is part of the exercise, and we have to inculcate that among our students and looking up at that. And I think we are in the country also, there are many things that can be done, and it's not always that you have to go outside in order to do things which are much more on a bigger scale. But through collaborations and all that, you have to use various tools. The demand is that you can't restrict to one kind of approach. So you should be able to sort of garnish a cooperation and be part of a bigger group to contribute your bit to that.
Right.
And I'm very hopeful that the next generation will do much better than the previous, and that's how history also proceeds, and I'm sure it will not be slowly just, but one day.
Thank you, ma'am, for your words of encouragement and for making time for us. It was a very, very interesting conversation. Thanks a lot.
You're welcome. Thank you.